Hi there. Welcome to this module where we will be looking at the two essential concepts, mitosis and meiosis. And we will be reviewing these two a little later once we have grasped some basic concepts. As we get started, I want to get us thinking about why this all matters. And often we end up looking at how a human being starts as a single cell, the fertilized egg cell, where the content from the sperm's cell's head part has fused with the egg cell's content. And what is truly amazing to me is that it is all from this one cell. We end up being such a complex organism with many specialized tissues, but it is all from just one cell. How amazing is that? But rather than looking at just the human development, as this course is all about the variety of life forms, I thought that why don't we look at that of the sea urchin? And what we see here again is that the life of this organism starts as a single cell. This divides into two, as seen here by this scanning electron microscopy image. And after just 4 rounds of cell division, there are already 16 cells. As these rounds of cell division continue, pretty soon we end up with a mature organism. And what is truly amazing to me is that here we see these complex, specialized structures of a mature sea urchin. Yet all of its varying different parts with such striking differences, all of those originate just from one cell. So that initial cell must hold instructions to all these different versions of cells that we see in a mature organism. And I guess that the next logical question is that why do we have cells dividing in general? Well, we can actually find two situations where the cell division must take place. And the division is very different in these two in terms of how it happens, especially as it becomes to how the genetic material is handled. And the first one that we are going to be looking at in meiosis is going to be the production of the egg or sperm cells. And these are known as gametes. So these are our sex cells. And the reason why this is such a special way of cell division is that to make an egg or sperm cell, we want to have only half of the genetic material in the resulting egg or sperm. This is because we get 50% of our DNA from the mother and another 50% from the father. Well, the other kind of cell division that we will see everywhere else in the body is going to be mitosis. In mitosis, the amount of DNA is 100% in all of the cells. And this kind of division is important, for example, for repairing any tissues that have gotten damaged or creating new tissues as they are needed. So this could be either as part of our growth and development or as part of just replacing cells that naturally die and get replaced over the life. So those are our two types of cell division that we will be looking at in this chapter. And we will actually start with mitosis, so cell division that takes place in all other cells in the body, with the exception of the sex cells, sperm and egg cells. These cells and how the genetic material in them divides is going to be the interest of ours in this first part of our videos. As we are getting started, I think it is a great idea to review some of the key terms that we must be familiar with. And we are going to start by noting here we see a cell and a nucleus inside it. And we might have seen this X or H-shaped structure before. And this is, of course, a chromosome. Chromosomes are DNA that has condensed and wrapped up into this X or H-shaped structure. And we see this during a particular stage of the cell cycle, a phase called metaphase. We will discuss these phases in a bit. Well, if we look into the chromosome structure and what it is made of, we will see that they are made of the chromatin fiber and nucleosomes. Chromatin is a mixture of DNA and protein, while nucleosomes are these bundles of protein around which the chromatin wraps up. So that's the structure of the chromosome, what it is made of. And finally, here we see, of course, the DNA, which is made of sugar phosphate backbone and nucleotides that connect the two strands of the sugar phosphates. You can think of the sugar phosphate backbone as these vertical sides of the ladder that we see here in red. So these are going to be two of those strands. And the bases are like these horizontal cross beams that we see on here when climbing on a ladder. And of course, all of this is then twisted around itself to give the appearance of the DNA that we are used to seeing. And the final term that I want to introduce is when we look at a section of a DNA, which codes for a particular protein, for example. Well, that part of the DNA is known as gene. I also want us to look at a single chromosome in a little bit more detail. So chromosome is made of two chromatids. You can think of these as these two sticks that are connected together somewhere roughly in the middle. And then we have also centromere. You can think of these as the part where the two chromatids connect together. One more thing that I want to discuss about regarding chromosomes is that certain traits are coded within certain locations of the chromatids. And we would always have this trait coded on both chromatids. This location where the trait is coded at is known as an allele. But do remember that it does not always have to be that what is coded in each chromatid, so each locus has to be the same. Let's look at that. So we can have a trait, and for example, let's say that is whether your hairline has a widow's peak, like former speaker of the House, Paul Ryan had. Notice this sharp triangular appearance of the hair right at the front. Or if you have a straight hairline, like soccer player David Beckham has. Notice how his hairline at the front is all straight, so no widow's peak. Well, we happen to know that the widow's peak is a dominant characteristic. So each individual has two genes for the appearance of the hairline, each coded in one chromatid. Because the widow's peak is a dominant characteristic, if the offspring inherits even just one allele from either parent with a gene for the widow's peak, this individual will always have a widow's peak. The other way how this can happen is, of course, by inheriting an allele for the widow's peak from both parents. So that's how you get widow's peak. The only way how one can have a straight hairline is by inheriting an allele for that. And remember that this was recessive characteristic, so it must be inherited from both parents, as we see here on yellow. So now that we know that we can have these different combinations of alleles to create either a straight hairline, which was inheriting recessive allele, so lowercase letter, from both parents. Or we can have a widow's peak. And this was possible by having inherited dominant allele, either from one parent or from both parents. Now let's look at what the names we use about these alleles. And here you can see that it is that if both of the alleles of an individual are the same, it is homozygous. And if the alleles are different, this is known as heterozygous. Now, before we get carried away with these capital and lowercase letters and inheriting things, which is not really a topic of this video, before that, I want to direct our attention back to the cell division process. So a cell cycle is a term that we use to describe the entire lifetime of a single cell, from when it first comes to existence to when it dies. And this cell cycle contains always various events which are regulated to take place in order and to be successfully completed before the cell can continue in its development to the next stage. So what we are going to do next is that we will look at each of these main stages which take place during a single cell's lifetime. Why don't we get going? So during G1 stage, known as G1 interphase, the cell grows in size. Its cell organelles increase in number. Once this has been successfully taken place, we are going to move on to S interphase. During this stage, the DNA within the cell is replicated so that now two sets of the DNA exist within the cell. Now we are ready to move on to the G2 interphase. At this point, the cell grows even more in size, and now it is going to start to get ready to divide into two. So at large, all of these stages focus on the growth and doubling the DNA of the cell. Now we are ready to move on to the stage called mitosis. And it is the stage during which the real cell division takes place. So the DNA and the nucleus get divided into two. This is what we call mitosis. And the division of the cytoplasm and the organelles within it, that is called cytokinesis. So at the end, the cell has divided into two. And that completes the life cycle of a cell. So that's the life cycle and its stages. And there is one more important thing that I want to add into this discussion. Have you noticed how this cell division process is so successful, rarely do we find mistakes in it. So it is really well maintained in check. And this is done by the cell cycle containing specific points where the progress gets stopped until we are sure that the goals of the previous stage have been met. These are known as checkpoints. So let's have a look at those. So the first checkpoint that we will have is known as G1 checkpoint. This is actually the most important check of all of these. And after that, the cell passes this checkpoint, it is now committed to the division. So during this stop, the DNA is going to get checked. And if there are any errors within the DNA, we have two options. The cell can try to fix the DNA, or the progress stops altogether. In the later option, the cell can just simply stop from progressing at all any further in its cell cycle. Then we are going to have also G2 checkpoint. At this point, the cell verifies that it is ready for the DNA replication, and if there are any issues with the DNA, we can try to fix it here too. The final checkpoint that we will see is going to be the mitotic checkpoint. To move onward from this point, all chromosomes within the cell must have attached to the spindle. And we will talk about what the spindle is in just a moment. So you can consider this as a way to make sure that everything has progressed the way we intend. And if there are any issues, well, then the cell would be stopped from moving ahead and making more of these faulty cells. I know that this may have seemed like a lot to take in. There are all these terms and events and so on. But this video really is something that I wanted to include just so that we have a good foundation to look at the next video, which is all about the real topic of this module. So next we will talk about mitosis. So I will see you in the next video, I hope.